Great. Good afternoon. Thanks very much for joining us for May's Maternal and Child Health Research Institute seminar series. May is Mother's Day, so we really focus on a maternal aspect of the maternal and child health interface. And we're really fortunate to have Mike Angelo, who's professor of pathology, and Kay Liao to basically present to us about this amazing new technology developed and applied to the maternal and child health interface. But first, before we get to that, a couple of housekeeping issues. Just wanted to thank again the MCHRI Education Committee, what's shown here, a wonderful group of people that really are helping us to provide up-to-date information to the maternal and child health community. So if you have any topics or questions or want to join the committee, please let us know. Next slide. I really wanted to, and as a demonstration of this really innovative approach, the committee has actually put together this fall's 5th annual MCHRI Symposium here at Stanford, which will really focus on community engaged research. We have really an all-star lineup of international experts who are going to really tell us about how to create an infrastructure of community engaged research to provide diversity education in our community, and getting information back and forth to really provide the benefits of academic results to the community and back and forth. So really look forward. Also a second topic on neurodevelopment and rising scientist research as usual. And so I wanted just to have you start looking forward to the call for abstracts for this amazing community engaged research based symposium. Next slide. And then next month, again, we're going to highlight the pediatric diabetes program here at Stanford, and Anna Gloin, professor of pediatrics and endocrinology, will be talking about insights into metabolic tissue development and function from human genetics, and with a small vignette from Nicole Krentz. Please don't miss this, because again, it'll be an amazing and informative seminar on June 2nd, so a month from today. But today we want to talk about the maternal fetal interface with Mike Angelo's group. Mike is a board certified pathologist and assistant professor in pathology. He's really a leader, innovator in the field of high dimensional imaging, expertise in tissue homeostasis, which you'll see in particular today, and was one of the inventors of MIBI, which is basically multiplex ion beam imaging time of flight, which allows many different data points on a tissue section to really then highlight in parallel what's going on in a very dynamic tissue. And today he'll talk about a normative template for vascular remodeling and immune tolerance with the student Kay Liao, who we'll introduce afterwards. So Mike, thanks so much for joining us. We're in for a big treat. And then as always, there's a Q&A session for those who have tuned in. Please put your questions in as you can think of them, and we'll have at the end a Q&A session where we can discuss the great results. So Mike, thanks for joining us. Take it away.
Great. You can see the slides okay?
Yes, perfect.
Okay, great. So yeah, thanks for having us today. I've got Kay with me here today. She's right here. I figured I'll start off, and then Kay is going to talk towards the end about some really cool stuff she's been doing in the area of mapping out the maternal fetal interface that has to do with some with sort of normative trends in glycosylation as pregnancy progresses in the first 20 weeks. And it actually synergizes really well with the stuff I'm going to talk about today. So I think that'll be really great. So just to kind of give you a quick summary of the presentation here, where my lab kind of started, it was developing this multiplex imaging platform for doing sort of high dimensional tissue analysis, which is called multiplexed ion beam imaging. And at the root of this was really wanting to sort of make the depth that we could interrogate human tissue biopsies sort of at the standard and the caliber that we can do with sort of single cell suspensions. And so I'll talk to you about that in terms of briefly about what that technology is, how it works, why we think there's some benefits with it. But then after that, I'd like to transition pretty quickly to, talking about the real meat of the presentation, which is this cohort that we have now completed. There's a preprint online now of how we use this technique called MIBI to be able to map out early changes in the first 6 to 20 weeks of human pregnancy. It's particularly with respect to what is the shift in sort of decidual into the state of decidual immune cells, and also what is going on with vascular remodeling in those first few weeks, since we know that seems to be a critical player in sort of healthy pregnancies and mitigating pregnancy complications like preeclampsia. But along with that, along with the MIBI study, I'd also like to talk about sort of how we're expanding sort of the suite of tools that we're now using to complement these multiplexed antibody-based imaging assays with some really cool technologies that have emerged recently for doing whole transcriptome analysis in these same tissue sections. And what Kay is going to talk about is how we've been able to extend imaging of tissue now to more sort of classical de novo mass spec imaging, and what that's done for us in being able to understand, sort of what Carolyn Bertozzi calls like the dark matter of biology, which is tissue glycans. So to start off with explaining multiplex ion beam imaging by time of flight, essentially this is just it's a modality for doing tissue imaging, an antibody-based tissue imaging assay. And so we use primary antibodies. And the way to think about it is, if you're familiar with immunofluorescence, it's sort of like immunofluorescence, except instead of using a light microscope, we're using an ion microscope. And instead of tagging the antibodies with fluorophores, which emit light when they're excited with a laser or a broad spectrum source, we're tagging the antibodies with mass reporters. And so if you're familiar with CyTOF, the tagging that's done to these antibodies is quite similar. So every primary antibody, we can select a staining panel to allow us to look at around 35 to 40 different proteins in the tissue at the same time. And so to do that, for every protein we want to image, we get an antibody that is reactive with that protein. And we tag that antibody with a metal reporter that usually is a isotopically enriched, like either transition metal or rare earth element. And so the rare earth elements, the lanthanides, if you remember just from chemistry, remember there was that block of like elements that was at the bottom, the 2 rows. So the lanthanides are the top row of those 2 rows. And so, and the reason those are really nice tags is because the natural abundance of these elements in tissue is really extremely low to non-existent. So that means you have a very nice quiet background when you're looking at these mass tags in tissue. And so we take the tissue section, it's mounted on a slide, and the tissue stained with all of the antibodies at one time. And so at that point, after the tissue staining, we wash off the excess antibodies. So the only things that are bound in principle are the ones that are bound to the protein of interest. And then we put it in the mass spectrometer. And inside that mass spectrometer, there's a stage where we can move around the sample and select the field that we want to image. And once we've gotten that field that we want to image, there's a primary ion source that produces charged xenon ions that are +1 charge. And those are accelerated around 30,000 volts to be focused down into the sample to a really small spot size on the order of about 1/4 micron or 1/2 micron. So down to about 250 nanometer resolution. Most of the work we do is around the 1/2 micron spot sizes. And so that beam is focused down. And if we've got an image that's sort of, you can think of as just being comprised of, just a grid of pixels, right? The beam goes to every pixel consecutively from left to right, right to left in a raster pattern. And for each pixel, it'll sit there on the pixel for say 100 microseconds to maybe a millisecond. And while the beam's sitting there, it's sputtering or eroding the sample and liberating the material that's a part of the tissue as the secondary ions. And the secondary ions contain those metal reporters that are on the antibodies. And those are transported to a time of flight mass spectrometer. And ultimately, by being able to quantify all of those reporters in every single pixel, we get these, nice sort of in-dimensional images where N is the number of antibodies that you have in the panel. And so and when you look at the images, it would, most of the time, it would be difficult to distinguish them from something like, 3-channel immunofluorescence, except instead of having 3 channels, you've got obviously a lot more than that. You can kind of toggle them on and off however you want to be able to display them. And so, getting the technology for this sort of established and stable was kind of the first step. And we've kind of transitioned within the last couple years, once we've removed this bottleneck on being able to acquire the data in a reasonable volume, to figuring out like how we're going to analyze it. So it's because essentially once you get up to the stage of having, lots of images that are 35, 40 channels per image, many of the ways that the image analysis was done for these types of data previously just doesn't scale well. And so we've had to go through and sort of, revisit how exactly can we come up with reliable computational pipelines to look at a lot of this stuff. So, our pipeline essentially begins with sort of low-level image extraction and noise removal and background removal. After that, we take those images and we perform cell segmentation. And so cell segmentation is just simply the process of running the images through software to be able to draw borders around each one of the cells in the image. And so we found when we first started off, a lot of these approaches really didn't generalize very well. If you had it in one tissue type, you might be able to dial in the parameters to kind of work well there. But as soon as you went to a new tissue type where the cells look different, the cell segmentation algorithms would usually perform poorly. So we worked together with a group at Caltech with the Van Valen Lab to come up with a deep learning method for being able to do whole cell segmentation. And this is kind of showing some examples here. And this actually performs very, very well across most of the major tissue types in the body. We trained it on over 1 million manual cell annotations across, I think, 15 different tissue types, 3 or 4 different modalities. So it's not even it's not just for MIBI either. You can use this for other fluorescence imaging platforms too. And we were able to show in that study that essentially, if you compare the output of this platform, which we call MCHRI, to the output from a human annotator, that essentially the performance between the two across different tissue types was pretty, there wasn't a statistically significant difference. So we have something now that we feel in most tissues can give us sort of human level performance for whole cell annotation, which has been a big game changer. It's shortened the amount of time that it takes us to identify cells from this bespoke process that we use in the beginning. It may take like, 2.5 months to something now where we can have this done in about a half a day. So once we've done that, we can actually start treating these data either at the cell level, and we can do stuff like cell clustering. So if you've ever dealt with any sort of single cell RNA-seq data or flow cytometry data, most of the methods that are used there are very similar to the ones we use here. We also look at things at the level of pixels as well. And the reason for that is that some features in the image don't really map well to sort of a cell by cell analysis. And so, for that, for instance, you could think about like extracellular matrix as one of those. And so we also have ways of being able to do pixel clustering to be able to define a lot of these extracellular features that aren't really assignable to single cells. And so once we've done that, we've done the cell segmentation and cell clustering, we can produce stuff like this called cell phenotype map, where every single cell in the image is colored by what type of cell it is. And from that, we can now go and identify cell neighborhoods, which is essentially reoccurring cell types that tend to occur together in the tissues. And so I won't spend too long on explaining exactly the algorithms for those for the sake of time, but this sort of completes, the sort of nested way that we have been looking at these images, which is at the level of pixels, the level of cells, and then at the level of sort of multicellular organization within confined areas within the tissue. So in the lab, I would say we've got, you know, the overarching goal really is we're trying to understand this relationship between tissue structure and phenotype. And, you know, about half of the stuff we do is in the realm of cancer, while the other half is focused more on trying to look at areas where it seems that there is conserved programs and pathways that are being recruited in either in a normative process or in pathology in a way that maybe is unexpected. And that sort of overlap is where we think that a lot of really useful information can come out. And so we've done work, for instance, in tuberculosis. And as I'm going to talk to you today about the maternal fetal interface. And this was kind of, you know, what people in the lab has referred to as my Angelo Lab tripod, which is, you know, how can we leverage things that we know about one field to learn about the other? And so, for example, you know, if you look at cancer, here's just a list of some of the things that we kind of think about when we think about aberrant processes allow cancer to persist, which is like immune suppression, which has become a really big one because of all the therapies that are coming out now that are post-directed, upregulation of endogenous retroviruses, angiolymphatic invasion, Warburg-like metabolism with aerobic glycolysis, vascular remodeling, ECM remodeling, and the genomic alterations. Well, the remarkable thing is that it's pretty well established that every single one of these happens in pregnancy, and particularly when you're looking at implantation of the placenta. And so, you know, sort of the conviction that we have is that this isn't happenstance. And what this most likely indicates is that many of the pathways, and particularly the coupling of these pathways that we observe in cancer, the reason that they are the way they are is because these are programs that are being usurped from human gestation. And so, like, for instance, if you look at, there are many instances where immune suppression is sort of coupled to angiogenesis and also to hypoxic signaling. And so it's, you know, when we see this over and over again of like coupling between these different aspects of cancer that sort of in a vacuum don't really make sense, but when you think about it in terms of establishment of the maternal fetal interface can make a lot of sense. And remarkably, we also see these things in tuberculosis. And so, you know, this has kind of been the big three areas that we focus in the lab so far. We're now starting to also look for sort of these reoccurring themes in other areas as well, as including HIV, preeclampsia, and endometriosis. And so we've published papers now on TB, a couple in breast cancer. And the one that I'm going to talk to you about today is the third leg of the tripod, which is, you know, and it's also the one that I think could be informing more than anything all the others, which is the first 20 weeks of gestation and the changes that are undergone inside the decidua. So the first 20 weeks of pregnancy is wild. It's really crazy stuff that goes on. You know, and one of the things that we know happens is the first thing is that there's this big shift at the maternal fetal interface towards a highly tolerogenic microenvironment that sets up sort of everything that's needed to maintain tolerance towards the fetus until labor. And one of the other processes that goes on that's sort of coupled and in concert with this is that with implantation of the placenta, you get the placenta does some remarkable things. So the placenta, which remember is genetically fetal, it has these finger-like projections called villi that grow into the decidua on the maternal side. And from the tips of those villi, that gives off the trophoblasts undergo differentiation to become extravillous trophoblasts. They take on a mesenchymal-like phenotype, so they go for an epithelial and mesenchymal change. They invade the decidua and they migrate around the spiral arteries, which start off looking like this. They're very small with a large amount of smooth muscle around them. By 20 weeks, these trophoblasts have participated in something called spiral artery remodeling, where they kind of seem to work hand in hand with maternal, sorry about that. My emojis came up. I don't know if you guys could see that. The seem to work hand in hand with the maternal NK cell subsets to remove the smooth muscle that's around these vessels. And at that point, they then invade the arteries and replace the maternal endothelium. So it's a crazy process. It really doesn't have much of a, I think, equivalent or precedent with anything else in human biology. And the reason why it goes under such, at least the reason it's thought that this extensive process of the vascular tree in the endometrium occurs is that it allows the vessels to get much, much bigger. And so on the right, you can see in a normal pregnancy, these spiral arteries, the lumens have gotten very, very large. And so this allows for a lot of blood flow to reach the placenta under low velocity conditions that probably prevent the placenta from being damaged and getting sheared off. And we do know there's a lot of evidence to show this process is sort of impaired and incomplete and many times in different variants of preeclampsia. And so, so the big goals that we had is that because this is in a human system and because of all the things having to do with pregnancy, you can see that this would be a kind of a difficult area to study in a lot of ways. And the issue with that is that there's many aspects of this that appear to be human specific, or at least primate specific. So being able to look at it directly in human tissue, we felt was really important just for understanding just getting a better idea of sort of decidual immune cells and sort of decidual remodeling and how that has to do with trophoblasts and what's special about that in primates in general, but also of how this could be used as a window for really trying to understand how these mechanisms are usurped in cancer as well. So the two goals that we had was really to define the structure and composition of human decidua from 6 to 20 weeks gestation. And really what we wanted to do was understand how these changes are correlated with kind of two axes. The first is the gestational age, so just how far along in time the pregnancy is. And the other is to understand if there's a sort of secondary coordinate axis there that is also driving some of these changes, which is what's going on locally around the spiral arteries and how does that relate to the EBT. So do we see that the decidua just changes in lockstep as time goes on, or as I think I'm going to be able to show you, is it that also with local tissue level regulation? So there's sort of like you see these ongoing trends, but then there's also some degree of local regulation as well. And so there are 3 people that have worked really hard on this study. This is Shirley Greenbaum, Aaron Soon, and Inna Averbach. And so in this study, we were able to work with pathologists at UCSF and we're able to go into the FFPE archives and get archival tissue blocks from women that had undergone elective termination between 6 to 20 weeks gestation. And from those tissue blocks, we were able to select around 66 patients where there was decidua decidua present within the tissue block. From those tissue blocks, then we constructed this tissue microarray, a couple of tissue microarrays actually. And those were, after that, we stained them with this 37-plex antibody panel. This primarily focused on really understanding EBT states as well as decidual immune cells. We then imaged it with MIBI, and we end up getting these images that look kind of like this. And so if I blow that up, you can see that this is showing in the inset here, you can see the output of Mesmer, which is it's able to sort of outline each one of these cells quite accurately. And so in red is vimentin, and that's the maternal fibroblasts, and then in green is CD14, and those are decidual macrophages. And so we did this for all the images, and then ultimately ran our cell clustering algorithms on them. And you can see now that we were able to sort of cluster out a lot of different immune cell subsets, trophoblast subsets, as well as major decidual structural cells like fibroblasts and glands. And so once we have those, we can map those back onto the tissue, and we can start to ask sort of at a cell composition level, a lot of questions about how are these things changing. To be able to augment that though, because we were really focused on spiral artery remodeling, this is another one of those things where the morphology of these changes don't really amend them, are really well suited for whole cell segmentation. So we actually took on a dedicated effort. This is the royal we, of course. Shirley and Aaron did a tremendous amount of work, and essentially in over 500 or 600 arteries that were in these tissue samples, they went through, and first we manually staged each one of these arteries to assess the degree that they had been remodeled. And we categorized, and they categorized every single artery as either stage 1, 2, 3, 4, or 5. And so I'm going to show you real quick what those different stages look like visually. So stage 1 you have these small unremodeled vessels. So you can see the endothelium, which is labeled as CD31, is intact. And they're also very small lumens, and you've got a thick layer of smooth muscle. With stage 2, you see these vessels are starting to get bigger. The endothelium is swollen a little bit, but the big change is that the smooth muscle has gotten disrupted and sort of fragmented. By stage 3, we see a few things. The first is that we now see that trophoblasts have invaded through the decidua to start to accumulate around the vessel. Smooth muscles also been lost more, and we see this continuous thinning out of the endothelium. By stage 4, this is kind of where things really start to get crazy. The EVTs invade the artery. Many times you find these whole plugs where they've occluded the artery completely, and those can stay for several weeks until around 20 weeks gestation. And you can also start to see these purple cells, which are the HLA-G positive cells. You can see here that they're starting to displace the maternal endothelium, and the vessel's gotten bigger. And then finally, with stage 5, the vessels are very, very large, and you've now gotten near complete loss of smooth muscle, and the majority of the maternal endothelium has also been displaced. So the first thing we did was, when we first looked at this, at these manual staging scores, we looked at it to say, okay, that was this first question, which is this highly concordant or somewhat concordant or discordant with gestational age? And I think it kind of fell into the middle option, which is to say that there's definitely a trend towards more remodeled arteries as you go from 6 to 20 weeks. But you'll see that early in gestation, we have these stage 4 arteries. And then in late gestation, you also had women that had some stage 2 arteries. So it's definitely there's definitely a correlation in the shift in terms of gestational age, but there's a lot of variance that we can't account for purely on gestational age. So that kind of reinforced this idea of what we were thinking, that like it is regulated in part by how far along in pregnancy are, but there's also these local factors that have to do with trophoblast invasion that seem to be at play too. So to really get a better idea of this, what we did was we took those manual stages along with a bunch of extracted features from these images that describe each of the aspects that were used in scoring these arteries. So like, how thick the muscle was, how fragmented it was. And we use that along with the machine learning approach called linear discriminant analysis to actually sort of create a trajectory of spiral artery remodeling that would allow us to give every single artery one number to sort of, quantify its artery remodeling score, to say where along this continuum it was. And so I won't dwell on this for the sake of time for too much, but essentially by doing that, we were able to, interpolate this trajectory and assign each one of these arteries, which are represented by a dot, to a single point along this trajectory. And so what that did for us was that now for every single image that we had, we were able to say, okay, if we look for each patient and we say, what is the average degree of remodeling for their arteries for a single patient versus their gestational age, right? We've got two numbers. We got a spiral artery remodeling score, and then we got gestational age. And we can now compare those with respect to all these other things, like for instance, what's the frequency of different NK cell subsets, how many EVTs are nearby, EVTs for extravillous trophoblasts. And we can ask which of those parameters seems to be able to explain better changes in these different cell subsets. And so by doing that, we were able to then see that the vast majority of maternal immune cells seem to vary pretty closely with maternal, with the gestational age. And so, in matter of fact, simply by taking these maternal immune cell subsets that you see on the right in J, Aaron soon was able to create a ridge regression model that would allow us to be able to predict really within about 10, 12 days accurately what the gestational age of that decidua was. So, so this is terribly interesting because, there's an overall shift that happens that goes away from a decidual NK rich environment, which is early on in gestation. And decidual NK cells go down very rapidly along with T cells. And what you're left with towards week 20 is a very macrophage rich environment. And these macrophages take on a heavy immune tolerogenic state that's CD206 and CD163 positive, as well as some other markers. And so, and what's really interesting, I think, for the context of other things like cancer, is this sort of gives a chronological trajectory to say how these things kind of occur and how they're coupled to other things that are going on, like ECM remodeling. And so the other thing that we noticed with gestational age is that it's been known for a long time that IDO is upregulated during pregnancy. As a matter of fact, I think some of the very early studies on IDO were in the context of pregnancy. And if you gave mice IDO inhibitors, they would are not able to carry pregnancies to term. And so we also saw this. We saw IDO expression was up, particularly in the glands. But what was very interesting was that this was highest early on, and then we found that by week 20, it sort of tapered off. And as that was going down, we saw that the endothelium within the blood vessels that were getting invaded by the EVTs, the IDO expression was going up. So IDO has sort of an immune suppressive effect. It promotes differentiation of T cells into Tregs. And so we see that kind of corresponds with when these genetically dissimilar EVTs are now getting back into now starting to come into a lot of contact with maternal blood as they invade the arteries. The other things that we saw was that there was this very strong prominent trend of like TIM3 and galectin-9 or macrophages sort of going up hand in hand. And so TIM3 and galectin-9 have also been implicated a lot in the context of cancer immunotherapy in terms of being another immune evasive pathway. So the last little thing I was going to show you in this study was there's sort of two models, thoughts for how spiral artery remodeling happens in humans. And, and obviously because it's in humans, it's been very difficult to kind of tease out which is which. And they really just differ on the path of invasion of the trophoblasts. And so the trophoblasts that, everybody agrees that they originate from the attaching villi. The difference is that there's some evidence that suggests that they could be. So on the left, let's start with this one. The trophoblasts invade the decidua, and then they come up to accumulate around the artery and then invade the artery from the outside. And so this pathway is called the intravasation pathway. But there's also some evidence to say that maybe that's not the whole story. Maybe what happens is they're actually extravasating into the arteries. And so extravasation is where the EVTs that are coming off these attaching villi, instead of going through the decidua to get to the arteries and invade, they're actually detaching and invading from the intravillous space directly into the arteries. And so this is called the extravasation pathway. So we're going to see if by kind of linking together, looking at trophoblast EVTs at each of the locations along these potential paths of invasion, if by looking at the frequency of trophoblasts and sort of what their phenotype looked like, if we could tease out which path looked like it was the more likely path of invasion. So to do that, we went back to some of these images and we really focused on EVTs and deeply characterizing the phenotype we saw by MIBI. And so so in the cell column or attaching villi, we had, EVTs here that we could characterize. And then if we go to the next slide, I can show you that, all of these purple cells are HLA-G positive trophoblasts. And so you can see some of them are far away from the artery, some of them are close. If they had invaded the decidua, we would call them interstitial. And if they were within like about 1 cell diameter of the artery, we refer to those EVTs as perivascular. And so the idea then is, is if we're looking at from the tips of the villi to within the decidua to around the artery to lastly, actually within the artery itself, if we quantify all these cells and we look at what's going on and what their phenotypes are, can we kind of piece together a timeline? And does that timeline look like it's more consistent with an intravasation process or an extravasation process? So the first thing that we saw, so spoiler, it looks to us, at least from the evidence that we have, not to say any of this is definitive, but all of the stuff that we see here seems to be most supportive of this intravasation pathway where it goes through the decidua and then into the arteries. And so the first thing that suggests that is if we look at arteries, if we look at how many which arteries have EVTs around them perivascularly versus inside of them or intravascular, what we see as we look at that as a function of how much smooth muscle has been lost of those arteries, we see that you almost always have perivascular EVTs here before you get intravascular EVTs. So they seem to be appearing on the scene. There's about a half of the smooth muscle's been lost, and we don't see the intravascular EVTs starting to go up until perivascular EVTs have already been there. So that's kind of the first thing. The second thing that we find is that endothelium, if we look at the loss of endothelium within the vessel, that doesn't seem to really matter that much for perivascular EVTs. So perivascular EVTs are sort of always there, irrespective of how much endothelial loss there is. But for intravascular, we see more and more intravascular EVTs as more and more endothelium is lost. So that to us seems to offer some support of the fact that breaks in the endothelium are necessary to get intravascular EVTs, which might mean that that's the direction that they're coming from. And then lastly, the other point that we were able to find is that if we look at sort of the phenotype of these cells going from the attaching villi or the cell column to the interstitial cells, perivascular, and then intravascular, there's this stepwise change in the phenotype of the EVTs, where they start off as highly proliferative with CD57 expression, and they transition to being bright for PD-L1 with CD56 expression. So all in all, together, we think that that kind of suggests that highly supports the intravasation mode of invasion. And so quickly, I'm just kind of breeze through this so I can hurry up and turn it over to Kay, and she can show you the coolest stuff. We've now gone and been able to dive in more deeply to look at each one of these trophoblast populations using nanostring DSP. And so essentially nanostring DSP, it's a way to be able to get whole transcriptome information from tissue, but being able to restrict where you read out transcriptomic information based on an immunofluorescence image. So with the marker panels that we saw of the MIBI stains, we then went through and using that in combination with where these EVTs were histologically in the tissue, we collected targeted ROIs to be able to get whole transcriptome data on EVTs that were each one of these locations. And so, we were able to do that for most of the cohort. And in doing so, we were able to define, we now see that there is actually very well-defined, nice differential expression patterns that we're seeing as EVTs go from attaching to these invading interstitial EVTs. And especially when we see there's some really fascinating changes we're seeing as the EVTs invade from the outside of the arteries inside, and that's what's being shown here. And so one of the other things that we're finding too is if you look at these transcriptional programs, many of the genes that we see are being recruited as these EVTs become more invasive are markers that have been shown in different studies in different cancers to promote invasiveness. And so again, we think that this is ultimately going to end up supporting the fact that invasive programs and tumors are probably templated in large degree off of gestational processes. And so now I'm going to hand it over to Kay, and she's going to talk to you about how we're, again, on these same tissues, on serial sections, we're able to go in now with a complementary platform to be able to do mass spec imaging to see what types of glycans are changing in each one of these compartments, particularly within the context of the extravillous trophoblast.
Thanks, Mike. So hi, my name is Kay, and I'm a graduate student in Mike's lab, and I've been studying glycosylation with MALDI mass spec imaging, which is a method that uses mass spec to visualize the spatial distribution of molecules based on their molecular mass. And a lot of my work has been done in collaboration with Richard Drake from Medical University of South Carolina, who has developed this method using MALDI imaging to interrogate glycans and map their spatial distribution in intact tissues. And glycans, just to say a few words, they are carbohydrate modifications on cell surface proteins, glycoproteins, glycolipids. And in proteins, they are one of the most common post-translational modifications that affect protein functions. They've been associated with cell invasion, immunosuppression, and I'll talk more about their functions later on. But here I just want to highlight that this method is great because it's completely compatible with FFP tissue, so no enrichment or derivatization is required. We use peptide N-glycosidase F, which is an enzyme that digests N-glycans. And N-glycans are a major class of glycans. They are attached to asparagine in proteins. And while there are other classes of glycans, there are no parallel enzymes that work as well for these other classes as pingase F works for N-glycans. And therefore, we are focusing on end glycosylation in this talk and in my study. And the application of this enzyme is by spraying. So the tissue is not immersed in buffer, and this limits the diffusion of the released glycans to give us spatial distribution. And the laser in the instrument then ablates the tissue at each spot. And this gives us down to 10 microns spatial resolution to visualize the glycans. So in my project, as Mike has nicely introduced, we are using the same decidua cohort that was analyzed by MIBI TOF in our preprint. And then we sprayed on pengase F to digest the M glycans and perform MALDI glycan imaging. And we obtained images at each glycan, which was identified by their mass based on carbohydrate composition. And so the goal of my study was to understand how glycosylation evolves across the first half of human pregnancy. And we think of it as we would like to use this normal model to understand how glycans are altered in, glycans are frequently altered in pregnancy complications and in tumors. And we think we can identify glycans as potential biomarkers for early detection of disease and to predict cancer severity. So diving into the results, we saw that there were trends in glycosylation that were correlating with gestational age. And so this is a representative image of a glycan that was increasing with gestational age. The tissue microarray is ordered by gestational weeks, and each core is a sample, and we can see that this is an intensity heat map where regions in red have higher intensities than regions in blue, and this glycan is enriched closer to week 20, and representing this in a box plot. On the y-axis is the intensity, and x-axis are the gestational weeks, and each point is a sample. And we can see again that this glycan is increasing with gestational age. So going a bit more into the structure of this glycan, we know that it belongs to a class of glycan called complex structures, which are characterized by branching. And each branch is comprises a disaccharide unit of glucnec and galactose. And so when there are 4 or more of these branches, we call them highly branched structures. And the addition of the 4th branch is driven by the action of a glycosyl transferase enzyme known as MGAT5. And it's known that many cancers increase MGAT5 expression, and these highly branched structures are one of the most common alterations in tumors compared to adjacent normal tissue. And highly branched structures have also been associated with increased invasiveness, and it's often thought that tumors upregulate these highly branched structures to promote invasion. So the main takeaway from this was that highly branched structures increase with gestational age. It's promoting invasion and associated with MGAT5 expression. And then Mike has also talked a lot about the placenta extravillous trophoblasts, which are fetal cells that are known to perform functions related to invasion. And now we ask the question whether these highly branched glycans could be expressed on these extravillous trophoblasts. And so what we did to answer this question was to correlate the glycosylation data with cell frequency data that was obtained from MIBI TOF. And so this is a Spearman correlation heat map between the cell frequency and glycan frequency across patient samples, where the rows are the cell populations and in the columns are glycans that were annotated by their mass peaks. And on top, they are color-coded with different structural features, such as whether they are silated, fucosylated, branching, and what class of glycan they belong to. And between each pair of glycan and cell population, we calculate the correlation coefficient, where positive correlations are colored in red and negative correlations in blue. And we saw that there was a group of glycans that correlated with fetal cells and extravillous trophoblasts. And many of these are silated and highly branched structures. And one example to highlight is this silated and highly branched glycan that correlated strongly with extravillous trophoblast frequency. In this scatter plot, the extravillous trophoblast frequency is on the y-axis and glycan intensity on the x-axis. Each point is a patient, and you can see that this glycan correlated significantly between with EVTs, suggesting that the trend that we saw in highly branched structures could be driven by extravillous trophoblast populations. So we wanted to confirm whether these glycans are indeed found on extravillous trophoblasts in the placenta. So we looked into this full tissue section to provide more spatial context. And this is a H&E stain where we annotated 3 main regions on the tissue based on histology of the placenta, namely the attaching villi, which is where the fetal villi attaches to the maternal decidua. And it's also, as Mike has mentioned, that they are enriched with extravillous trophoblasts. And we also annotated the fetal villi regions, which are where cytotrophoblasts are found, and the decidua regions where maternal stromal cells are found. And so then we perform multi-glycan imaging on the full tissue section. And we ask the question of like where these highly branched structures are located. And again, this is an intensity heat map of the glycan image where regions in red have higher intensities of the glycan and regions in blue have lower intensities. Zooming into this smaller region here, just to illustrate that we saw this glycan was localizing to the attaching villi and decidua regions, and there was lower levels of these glycan in the fetal villi regions. So this confirms that highly branched structures were enriched in regions where extravillous trophoblasts are found. Then we perform unsupervised clustering of pixels from the full glycan images. And each color here represents a cluster of glycans. Again, zooming into this region, the orange cluster is enriched with highly branched glycans, and it's co-localizing with where the anchoring villi and decidua are. So this was really exciting to us because it confirms that the highly branched structures are basically localizing to the attaching villi and the decidua, which are also where extravillous trophoblasts are mostly enriched. So now going to some like ongoing work where we are looking at co-registration of multiplex IHC image with MALDI, and we want to directly associate extravillous trophoblast staining with images of highly branched glycans. So this is an immunofluorescence staining of extravillous trophoblasts using HLA-G as a lineage marker. And on the same tissue section, we perform multi-glycan imaging, and these are some representative image of highly branched glycans. And you can see that these glycans have very similar structures except for slight variations in fucosylation and silation or further extension of the branches, but they are all localizing where the extravillous trophoblasts stain positive. So now we are looking into methods to perform pixel level co-registration and to further infer functional relevance of these branched glycans co-localizing with extravillous trophoblasts. And now I'll pass it back to Mike to say a few concluding words.
Yeah, so the only other thing I was going to say to highlight is that, we're spending a lot of time really understanding how you can meaningfully sort of leverage each one of these techniques to identify things that would have been difficult to sort of ascertain otherwise. And so, I kind of briefly alluded to the fact that by looking at nanostring in combination with the MIBI data, we're sort of able to discern what EVT populations are being differentially expressed depending on their context. Then, as Kay just showed you, with the MALDI images, likewise, we're able to correlate this with the MIBI study to be able to figure out what EVT glycans are present. And so the cool thing is that now we're really starting to see how we can close the loop between these two things. And so, yeah, I think Kay alluded to earlier, that we have, that she found this really interesting correlation with increasing levels of a glycan that's produced by an enzyme called MGAT5. And so that pathway you can see is here. And so by seeing this, we've now gone back and we've looked again at EVTs within each one of these spatial compartments and looking at the nanostring data to sort of get insights into what these enzymes are doing. We kind of use this as a test case, and we're able to find out that, essentially we can see progressive increases in MGAT5 expression as we go from attaching villi to these invading interstitial ones. And so this is probably the thing that we're most excited about, I think, is the fact that we're going to be able to, link together these sort of very different data sets to be able to, do different types of correlation analyses that kind of wouldn't be possible with just one or the other. So that's it for us. If there's any time, I'm happy to take any questions.
Awesome. Mike and Kay, that was amazing. Thanks so much for that introduction to the technology and for this incredibly exciting finding. If you want to put questions in the Q&A, I'll start off with about 7 or 8 minutes. The first one actually was, so you linked it to from Robbie Parkman, which is you linked cancer and the maternal fetal influence. What about tissue tolerance, graft versus host disease? Have you looked? And is that also a model for the same type of immune tolerance?
Yeah, I know it's a great question. So I think, I think the other areas that could be a good model for sort of recruitment of sort of gestational like tolerance could be like what makes solid organ transplantations successful in some patients and not in others. I think there's probably going to be an overlap there. In terms of graft versus host, that's a good question, because it's sort of, in that case, it would seem almost like a failure to recruit these types of processes.
Right.
Yeah. And I think that could also be, I think any of these that involve like allografts could be very, very interesting to look at. I mean, it's essentially what it is. The fetus is an allograft. And so, and so I think being able to kind of, look for these similarities, it gives us a timeline to kind of template things against, where in all these other diseases, I feel like we're just getting snapshots, but we don't really know like how we got there.
Exactly. And then the obvious question you're begging is, are there blocking antibodies to these branched glycans that might be useful for immune checkpoint therapy for cancer, or to prevent or to induce tolerance for fetal loss, for example?
Yeah, I'm sure you could make blocking antibodies. I think the thing that is a big unknown is that is going to be, and Kay, chime in here if you don't agree, because Kay knows way more about glycans than I do. But I think I wonder if we're going to be able to find glycan structures with a high degree of specificity to pregnancy and tumors, because with all of these, I think the big question is always, it's like, okay, these things are recruited here, but like, do they happen to be in your heart too? I mean, it's sort of, so I think that's kind of an unknown at this point. Is that I think this will give us a lot of mechanistic insight, but at the same time, I think it's still an open question how orthogonal these things will be with normal tissue homeostasis.
Right. And are the cells of origin are expressing MGAT5 or the glycans, the branch glycans? Is that the do you know the cell type that's expressing those? Is it a specific cell type, or
we suspecting it's EVTs? Yeah.
I think we also see the expression in stromal cells, and they could play opposing functions, which is another very interesting. I don't know we can say about that, but it's just like really interesting to see that different cell types can upregulate this MGAT5 and like have different roles to play depending on where these cells are. And I think this brings in again like the importance of looking at the spatial distribution of where the cells are
and what protein they're on.
Yeah,
Is it,
yeah.
right. I mean, that may be what's underlying the differential function, is that in different cell types, you know, these same glycan modifications may have opposing effects depending on what protein they're on. And so I think that,
Right.
and I think that, and the thing that I think is good for this is that, you know, we're not seeing, the thing that's very interesting, I think, from the sort of cancer standpoint is that the glycans, it's not a infinite universe, right? It seems to be a pretty,
For in glycans.
for in glycans and decidua, right? It seems to be a fairly reoccurring group of players that we're seeing in there.
That really kind of like reminds us of how it looks, or like what people have seen in tumor samples. But now we have like a timeline to track how it progresses.
Right. Yeah. And then a follow-up question is, are these branch glycans, complex glycans, correlating with the spiral artery remodeling compared to the gestational age? Because you talked about gestational age, but does that correlate as well?
Well, that TBD, I think at this point, I think we still need to look. Yeah, we haven't we haven't delved in that deeply yet, but we're going to see what's I think if they're correlated with EVTs, they're probably going to be correlated because EVTs from this paper we know are like heavily correlated with spiral artery remodeling. So preferentially, so I would suspect at least a subset of them are correlated with spiral artery remodeling.
Right. And then the big band that you're seeing right at the interface. So walk us through your model for what, I mean, like your hypothesis for what that's doing. Is that the immune tolerance function? Is it immune tolerance? Is it you know, is it invasion map? I mean, what is it? What do you think the function is?
The band, what, I'm sorry, which one are you referring to?
The big band of the complex glycans right at the interface between the trophoblasts.
Oh, yeah, I don't know. I mean, it's sort of it.
I mean, it's pretty noticeable. I mean, it's massive. Yeah. It's like it's just sitting out there.
Yeah, I mean,
very interesting place.
It's you know, it's that place. You know, I'm wondering if there's going to be a lot of sort of synergy that's going back, because you know, right at the basal plate, you also get heavy stiffening of the ECM, which also promotes it. You know, and there's different receptors and a lot of these ECM. So I don't know. I'm wondering if it's somehow sort of working in tandem to promote and, you know, to help promote invasion, but it could also be promoting tolerance. You know, I think it could be either.
Okay. We're trying to get you to speculate wildly, but and then from the rest of the community, in terms of collaboration with your lab and do you do maybe just for humans, or is this also available for mouse models and you know, just sort of a general kind of lab advertisement for the community about in terms of interacting with you?
Yeah, no, I mean, we can do, and if an antibody works by IHC, it'll usually work on MIBI. I mean, so it's sort of, you know, we've spent most of the infrastructure we've accumulated for reagents is human, but I mean, you know, there's no reason we can't do mouse studies. I mean, in some ways, it's probably a larger degree. You know, with mouse, you've got a lot more control over tissue preservation,
right?
Which can be a big help. I mean, because, you know, we've kind of built everything out so that we're just compatible with FFPE, but, you know, with mouse, you can do frozens and you can do all these other things. And so, no, your mouse tissues, yeah, we can, anything with antibodies pretty much. Great.
And we have time for one last question, which is, there are other spatial localization techniques like Visium and Codex, and how's the resolution using the image processing that you've done, which is amazing, compare with the current state with Codex and Visium and those sorts of analysis for spatial expression studies?
Well, I mean, Visium's pretty chunky, you know. I mean, Visium's right? It's like, I think like 100 micron or
No,
something
it's,
like that.
I mean, it's bad, but I just was.
Well, it's not bad. It is what it is. It is what it is. I mean, I think the big thing is here, and I think this is probably a good talking point, is that, and particularly, it's a disease that's kind of enveloped Boston and the Bay Area, which is this single cell disease, right? Which is that everything needs to be single cell. And I would strongly argue that, like, you need one of your assays to be single cell resolution, right? But like, if you can get down to things, feature sets, like for instance, I think like if you have subcellular data on a serial section in the same section, and then you do Visium on it, that's probably plenty of information to figure out what you need to figure out, right? And I think like, and for us, we've been using nanostring because the reason that we like nanostring is that you can select ROIs based on cell, on a specific cell population. So we can look and say, we only want to get HLA-G positive cells, and we can collect them from there. But I think for all of these tools, the main thing I would encourage people to do if they're just getting into it is don't get caught up in the trap that like every single thing you do has to be single cell, right? Because it imposes logistical complications that make things pointlessly much harder than they have to be. We found that like simply by doing adjacent, like there's a lot of people are like, what if you could do the assay all in one section? And we're like, well, I mean, you could, but it's like, the sections are only 4 microns thick, right? And so it's like, in the resolution of most of these modalities is tens of microns. So it's sort of the axial separation is actually smaller than the lateral, right? So it's sort of, I think that, the transcriptomic, the resolution of most of them, I think is already pretty good, and so, yeah, I don't know if that answered your question, but
It did very much so. Thank you very much. Let's see, anybody else? Last question, do cells change their glycan phenotypes as gestation approaches labor?
No, yeah, I don't know. Probably.
Well,
Well, we could check. I mean, actually, term placenta is probably the easiest to check, right? We could definitely look.
I think what you have to do is call up David Stevenson and work with his lab and Bryce and then answer that question with him. It was his. So I suggest that you email him and he'll has the tissues and to basically provide that for you. So that'd be a great question to work together with.
Yeah, absolutely.
Okay, David, you heard him. He said yes. Okay. Anyway, Mike, Kay, thanks so much for a phenomenal seminar. And we'll look forward to much more and interesting interactions with the maternal and fetal interface. So thanks very much.
Yeah, thank you.
Thank you.
Have a
Take
good one.
care.
Bye-bye.
Bye.